Good morning, students. This is a continuation of part 1, sexual reproduction in the androecium. Okay. So the pre-fertilization. So I already explained you pre-fertilization means in this pre-fertilization condition there is the fusion of male and female gamete. It takes place. The fusion, it has to fuse. Before that, it has to produce. What it has to produce? It has to produce the male and female gamete has to be produced. So where the male gamete of the plant is produced, it is produced in the androecium. And female gamete, where it will be produced, it is producing in the ovule. So I am coming to the pre-fertilization stages, that is the male reproductive part, androecium only I am going to explain you today. The structure and parts of the androecium, that is stamen consisting of anther and filament, I had explained you in the last video part 1. This video explains the first how the flower is coming. So this diagram that's showing the development of flower, how the flower is first get developed. Hormonal and structural changes in the plant leads to differentiation and development of floral primordium. Yes, floral primordium means there is a group of cells or the tissues which is bring out the flower is called the floral primordium. So if you see this diagram, this is the buds which is showing, that is a group of cells or the tissues which is showing here. This bud is its bud like structure. This bud like structure, the group of tissues will bringing the future pistil, and this horn like structure will bringing you the sepals, and this is the stamen which is bringing androecium part of the flower, and this is bringing the petals, that is calyx. So and this is bringing the pedicel, that is a stalk of the flower. So these all the developmental stage, that is your development of the flower, you have studied in the last lesson, tissue and tissue system, in the last year itself. Okay. Next I am coming to the male reproductive part is androecium, female reproductive part is gynecium. Today I am explaining only the androecium. Androecium structure and its parts I had explained you in the last video. Now the male reproductive part of the androecium, we are going to see only development of anther, microsporogenesis and transverse section of mature anther. So the transverse section of mature anther, they will be asking you in the most of the question paper, they will be asking you transverse cut section or the transverse section of the mature anther. Microspore and pollen grains and development of male gametopy. Today we will see first 3 topics only. First I will move on to the this thing, that is development of anther. This diagram is your textbook diagram. I will explain you this from that. So it is easy only. So there is a homogeneous mass of cells which is just surrounded by a epidermis. So epidermis is a layer which covering the homogeneous mass. What is a homogeneous mass? You may ask the question. Homogeneous means that is similarly all the cells will be same size and same structure. It is called a homogeneous mass. Homogeneous mass means homogeneous, same size and structure of the cells will be linked or the grouped, and which is covered by a single epidermis is called anther primordium. This is first stage growing, that is young development, that is a young development of anther takes place. Next stage is differentiation. What is the differentiation? This homogeneous mass will become somewhat, all the cells is presenting inside of the epidermis is get enlarged with the conspicuous nucleus. That is conspicuous meaning is nothing but well clear nucleus you will be getting an enlarging of these cells. So this is called differentiation of archisporial cell from the anther primordium. So next stage is I am explaining it. There is a somewhat butterfly shape. That is, I am telling it is a butterfly. It is butterfly shape. So this archisporial cells is undergoing a periclinal division, anticlinal division. What is a periclinal division, anticlinal division? Periclinal division means nothing but there is in the cell or the tissues getting divided by parallelly. It is getting divided, all the cells which is getting parallelly divided, and it is giving the parietal cell. What are parietal cells and sporogenous cells? It giving a 2 types of cells in the divisions, periclinal division. Periclinal divisions means all the cells is parallelly get divided and giving a 2 parietal cells and sporogenous cells. This sporogenous cells is always going inside towards the center, and the parietal cells is also always moving to the periphery of the anther. Periphery means outer layer of the anther. So parietal cells will giving the anther walls, anther will be having these following walls. These walls will be brought by our parietal cells. And sporogenous will give the inside the pollen grains. That is, pollen grains will be presenting inside the anther cavity, will be filled with pollen grains. So these pollen grains will be having the this thing. So this is pollen grain is given by sporogenous cells, and parietal cells is given by our this thing only, that is given in the anther wall layers. And formation of wall layers. What are all the wall layers? That is, you can see in the this thing. So this is in this diagram will explain you nicely. This is sporogenous stage. What is a sporogenous stage? From this stage only pollens will be getting developed inside the anther. That is sporogenous stage. You see here, that is first one is this light color is epidermis. Next yellow color layer is middle layer. Next is tapetum. Tapetum means that is inner part. Okay, next is into that middle layer. Next is tapetum. Next to is the you see here it is epidermis. Endothelium is a red color layer and yellow color layer is middle layer, and inner one blue is tapetum, and small yellow color dots are pollen grains. And this is the structure which is presenting in connecting of the two lobes is called the stomium. Okay, so these are all the four walls layer, four layers of the wall of the anther which is coming from the parietal cell. This parietal cell is nothing but periclinal divisions of the archisporial cells. Okay, so sporogenous cells, what it will do, man? You may ask the question. The sporogenous cells is sporogenous cells also getting divided by it is mitosis. Is the sporogenous cells which is undergo the mitosis process. So thereby it producing the microspore mother cells. Microspore mother cell. It produces the microspore mother cell. See here. So it undergoes the this sporogenous cells is giving the pollen. It is giving the pollen mother cell. That is it is undergoes the mitosis process, so that it is giving a microspore mother cell. From the microspore mother cell or the pollen mother cell, this mother cell will be having the diploid chromosome. It is undergoing a meiosis process. It is giving the what is that? That is, it is giving the pollen grains. Okay, so pollen grains is coming from the microspore mother cell or the pollen mother cell. So in the sporogenous tissues is undergoing a mitosis process. At the end of the mitosis process, we are getting the pollen mother cell or the microspore mother cell. From the microspore mother cell, it is a diploid in the nature, but it is undergoing a meiosis process. It is giving the pollens. That is, pollens mean tetrad pollens. What is tetrad pollens? In one thing, that is, the pollen will be there, but pollen will be arranged in the 4-4. So it is called tetrad of pollen. Soon it is get separated and it is become as an pollen grains. So pollen grains is formed in the microsporogenesis of the androsium, and the anther wall is produced in the development of anther. So you all hope I understand that you will be interested in this thing. Next, I am coming to this. So this is sporogenous tissue which is undergoing the sporogenous cells undergoes first is undergoing the mitosis process. The mitosis process, the sporogenous cells or the sporogenous tissues is completely, it's become a last generation of the sporogenous cells division is become a microspore mother cell. Microspore mother cell will follow the meiosis, so thereby it is giving the microspore in the tetrad in the stage. Soon this tetrad will be separated as the microspores. This tetrad will be separated as the microspores. This microspore is in the young stage, it is called microspore. If it is mature, it is called pollen grains. So pollen grains is coming from the microsporogenesis process through the sporogenous cells. Sporogenous cells is coming from the that is parallel division of the archisporial cells, which is towards the center of the anther. That is in the paraclinal divisions also give the parietal cells, which is towards the periphery of the anther. So what I told you, a homogeneous mass of cells surrounded by epidermis. The archisporial cells is divided by periclinal divisions to form the primary parietal cells towards the epidermis, and primary sporogenous cells will give the towards the inner side. So sporogenous cells is giving rise to a microspore, and it is matured as the pollen grains, and these periclinal divisions will give you the this thing, that is your parietal cells is giving a anther of the anther wall it is giving. So this is just microsporogenesis diagram I explained you. This is your anther, this is your stamen, this is a cut section of the anther. So this is the showing the connective tissue. The middle, that is called connective tissue. It is nothing but it is a sterile tissue, that's it. Then this is 4 is called pollen sac, and this layer will be lined by 4 walls. That is I explained you here. Here you can see, that is first layer of the anther is mature anther is showing the first one is epidermis, next the violet color layer, next layer red color layer is endothecium. Don't use that color, this is I am telling you for the understanding purpose only, that's all. Next I am coming to the middle layer. Next is tapetum. Next is tapetum is innermost blue color layer, and before the innermost layer you can appreciate some yellow color layer is called middle layer, and this is the stomium. Stomium means the endothecium layer will be lacking in between the junction of the two lobes is called the stomium. This is help for the dehiscence of the pollen trains. Dehiscence means the splitting of the anther so that the pollen trains will be coming out for the pollination. So this is your star, the structure is called a pollen train. So other sporogenous tissue, again you know sporogenous cells is coming from the periclinal division of archosporia cells. So the sporogenous cells is coming out is following the mitosis process, so that you are getting the pollen mother cell or the microspore mother cell. Microspore mother cell again following a meiosis process, so that it is giving a tetrad of the microspore. This tetrad of the microspore or the pollen soon get separated into the separate trains is called as the pollen trains. So this is just theory part. 4 haploid microspore is coming from the microspore tetrad, or the 4 haploid microspore soon it is separated, which is coming from the microspore mother cell. And microsporogenesis term meaning as formation and differentiation of microspore is called the microsporogenesis. This is 2 mark question they may ask you. What is a microsporogenesis? Formation and differentiation of microspore. Because microspore is produced here as tetrad form. Then it is converted into a separated. So it is produced and it is formated and it is differentiated. So it is called the microsporogenesis from the sporogenous cells. Next is pollinium. What is called pollinium? If there is a chance in some plants like a calotropis. Calotropis, you know that is a in Tamil it is calling as Erukum chedi. Okay, this calotropis will be, there is no separation in the tetrad of the microspore. It is only, it is fused along with the 4. So that is called as an pollinium. And this pollinium is somewhat look like a camp-like, clip-like structure. So it is called corpusculum. Okay, there is in some of the long projection filament is coming out from this corpusculum. In the case of in the plant of the calotropis is called retinaculum. So the whole structure of the calotropis is look like a inverted letter of the Y. So is called a translator. You should know the 3 terms. That is, one is a pollinium. What is called pollinium? Instead of there is a no separation of the microspore, is it is tetrad in the nature always. It is not get separating, is called pollinium. So in that pollinium we can appreciate in the calotropis. So the calotropis you can see some camp-like or the clip-like structures of the microspore which is not at all getting separated. So that is called corpusculum. That corpusculum having some long projection from the microspore is called the retinaculum. So the whole structure is looking like a inverted letter Y is called translator. Okay. This is just sporogenous tissue or the sporogenous cells. That is, sporogenous cells is a two diploid in the nature, so that it is producing a haploid microspore, and microspore tetrad is pollen or the haploid in the nature. So it is become, this microspore is soon separated, and it is in the young stage it is called microspore, and it is developing as the pollen train. Okay. Next in this thing, anther wall layers. That is, anther wall layer is, it is made up of the epidermis, endothecium, middle layer, tapetum, sporogenous tissue. I explained you. Okay. So this epidermis is this is violet color thing I am telling here. So you see this diagram. This is a one single layer that is protecting the whole anther along with the pollen trains with the single layer of the cells, and these epidermal layer cells also undergoing the divisions. That is, it undergoing the divisions, mitotic divisions, and it is giving the it is growing. This epidermis also growing along with the internal tissues of the anther. So internal tissues, that is, you know, that this tapetum layer, middle layer, endothelium layer, all the layer also getting developing day by day. It is has to be developed. So it has to be developed, only it can accommodate the more pollen. So along with the inner, these all inner tissues has to be developed. So once it is got developed, your epidermis will be getting developed. Once it is getting developed, along with that, this internal tissues, the epidermis, single layer of the epidermis also getting developed. Next there is, and the function is dehiscence of the anther. It is helping for the dehiscence of anther. It has to be open to release the pollen. Okay, this pollen grains. Stomia means present between the two pollen sacs. Stomia means it is present in the two pollen sac. There is an absence of the endothelium. In that place, that is somewhat is thickened by the epidermis, is called the stomium. It is also helping for the dehiscence to release the our pollen grains. Next is middle layer. Next is endothelium layer. So next is, in between that there is, and the endothelium layer is there. In this endothelium layer is nothing but there is it is also single layer, this red color layer, but it is not a single layer. It is a radially elongated layer. Radially means it is somewhat sharpened cells. So radially elongated. Radially means it's somewhat sharpened layer. So, and its inner tangential walls. Inner tangential, I had explained you in the last year itself. They are in tangential, and it's a radially elongated layer. This layer is producing alpha cellulose substances. That also alpha cellulose substances is used to absorb the moisture from the air. Air will be having some moisture that will be absorbed by the cellulose, which is produced by the this inner tangential wall layer of our endothelium. So this absorbing the air from the moisture from the air is called hygroscopic nature of the alpha cell substances, of alpha cellulose substances produced by hygroscopic nature of this and endothelium. Endothelium is nothing but inner tangential wall. That is, that inner tangential wall will produces the alpha celluloses. So this and inner tangential wall is nothing but endothelium. Inner tangential cells will be producing alpha cellulose that have the tendency to absorb the moisture from the air. So that is the nature of the endothelium. So in some of the plants, the endothelium will be absent. That is example, that is our aquatic plants will be absent in some case of the aquatic plants. Next, middle layer. Middle layer is, it is 2 or 3 layer will be, that is in the yellow color is the middle layer. It's just epidermal cells will be present in the middle layer. This is nothing but this epidermal cell will be lysed or the degenerated once the anthers get matured. That's about their middle layer. Next, I am coming to the tapetum is the innermost layer. Innermost layer means you should know this is all the blue color is here, innermost layer of the tapetum. Tapetum is very important part of your anther. So this diagram is very important. They will be asking you always in the 3-mark question. Is many of equally all midterm, all the question paper is coming along with this question. This is very important. You should not leave any part of this diagram. So this tapetum is nothing but innermost layer of the anther. It is dual in origin. How it is originated? It is partly originated from the peripheral layer of the epidermis, and partly originate from the connective tissue, this somewhat flower like. From this and from the epidermis it is originating. What is that? That is our tapetum, innermost layer. So, and it is a dual origin. It nourishes the developing microscope. The microspore which is getting developed, by the or the nourished by this tapetum. And there is a sporopollenin, pollen kit, tripinin. The other number of proteins also produced by or contributed by the tapetum. So this tapetum is very important for the incompatibility reaction. Incompatibility means whether the pollen have to fertilize with some other egg or not, whether it is suited for that, whether it will give the fertilization or not, that will be determined by the incompatibility reaction. So that incompatibility reaction will be given by the sporopollenin, pollen kit, tripinin of the tapetum. And also it's control the fertility and sterility of the pollen. And there is 2 types of the tapetum will be there. One is one is in the secretory tapetum. Secretory tapetum means it's also known as parietal or the cellular tapetum. This tapetum is used to maintain the integrity and position of the cell wherever it is present in the anther. Where different types of cells you seen in the epidermis, in the middle layer, in the endothelium, in the inner tangential wall, all the cells has to be maintained their integrity and their original position maintained by the secretory tapetum. Whereas the invasive tapetum means it is somewhat loosely arranged tapetum. In this type of tapetum, cells will be fused. Their periplasm of this invasive tapetum layer cells will be fused together. It is called as a periplasmoidal in the type. Next, functions of the tapetum. It is used to nutrate the young microspore. Not only the microspore, developing microspore mother cells, sporogenous tissue. All the sporogenous tissue we saw in the anther layer. So all will be developed by, all will be nutrited by our tapetum only. So Ubisque bodies means the terms, there is the granular structures which is present in the tapetum, and through this bodies only it is giving the somewhat roughness to the pollen wall surface. If you see the pollen you are taking in your hand, you may see some roughness. So that is roughness of the surface will be given by the Ubisque bodies of the sporopollenin. Sporopollenin is a part of the wall material. That wall material will be given by the Ubisque bodies of the tapetum. Pollen kit also one of the wall material which is also giving the roughness to the pollen surface. Exine is a protein. That protein I will explain you detailedly in the next class, which is also used for the rejection reaction of the stigma. Whether this pollen is very ready and get ready, can able to fertilize the stigma or not, that will be judged by this exine protein of the tapetum. Okay. This is just overall. Anther is consisting of epidermis and archisporium. Epidermis, inside the epidermis, the archisporium is present. It is a parietal tissue, and sporogenous tissue is coming as a pollen microspore. Next, pollen tetrad, pollen is getting ready. And endothelium, middle layer, tapetum. Tapetum consists of inner tapetum, that is a innermost layer, dual in origin, originating from the connective tissue, as well as this epidermis layer. It has two types, invasive types as well as the secretory type, protecting cellular integrity and this thing. Middle layer is just epidermal cells. Endothelium intertangential, hygroscopic in the nature. Okay. So this is the epidermis layer, epidermal homogeneous mass surrounded by epidermis coming as the this thing, and differentiation as a archisporial cell. Archisporial cells is become a parietal, simple periclinal division parietal, and sporogenous cells, and sporogenous is become a pollen grains, and the parietal is become an anther wall. Okay, hope you all understand this video. In the next video I will be explaining of the development of male gametophyte. Thank you.